Oh, oh, my sound electro. Hi ha. Hi ha. Hi ha. Hi ha. Okay, let's get our slide ready. First, I need to. What sound electric? Hi. Never mind. Background music a bit soft. Oh, Pinocchio. Yeah, we good. Okay. We need electric sound. So much electric sound. I don't know. Sound. Hi. Hi. Okay. Yeah, the sound okay. Let's double check here. More of my monitor, so I won't hear myself. Better off. Okay. It is what it is. Okay, so welcome to photosynthesis, where I will share about the light reaction and dark reaction in photosynthesis. Okay, in photosynthesis topic, there are not light and dark reaction only. There are type of photosynthesis. There are biochemistry, biomolecule of photosynthesis, which involve the deeper topic about this light and dark reaction. So, what is photosynthesis? Maybe we know. Okay, the plants. Okay, they eat carbon dioxide and then they give up oxygen. Okay, this is a general knowledge for like primary school. They learn about science. They learn about plants. Okay, this is the not the very first thing, but they know what is this. Hello Meow, welcome to the stream. 欢迎。Okay, today we are going to discuss about photosynthesis 光合作用。逐渐安安。Okay. Okay, so back to the topic. Primary school know this, but it's a very general knowledge they know is plant do photosynthesis and give out oxygen. It's not a primary purpose of the plant to produce oxygen for us. Plants are not that kind. Okay, so the main reason they do is to make their own food, just like the thumbnail, just like the poster, whatever stated. Yeah. So let's dive to the first page. Okay, so in this stream, this lecture, this tuition class, this whatever, the learning objective will be the general concept of dark and light reaction. I cannot open my lamp, never mind. It's not charged. Okay, so this is the two, and then I will share two mini experiments related to this reaction, light and dark reaction. Somehow related, okay, supposedly. So it's a very interesting experiment. Yes. No S grammar wrong. Okay, GG. Oh, okay. So where does plant do or carry out photosynthesis? As we know, is in the chloroplasts of cell, which is the green tiny button in plant cell. Very important. Okay, so in chloroplasts there is some pigment in chloroplast, which is called chlorophyll, okay. So this chlorophyll is the pigment, okay. Chloroplast is this whole thing, including outer membrane, inner membrane, thylakoid, stroma, okay. So you have to differentiate very well about these two, chloroplast and chlorophyll. Chloroplast is the organelle and chlorophyll is the pigment. Okay, so two types of photosynthesis reaction. In this part, I will be discussing about the light reaction and dark reaction. So it's very deep, like okay, dark reaction, okay, light reaction, what is that, okay. So in a very simple words, light reaction is energy generator, okay. They use sun and everything, and this process need light, need the presence of light to carry out this function. No light, no light reaction, okay. In other words, this light reaction is called light dependent reaction, okay. It depends on light, so it creates many energy molecule, which I will show later. Energy generator, okay. Remember, the energy molecule in biology is called ATP, okay. Adenosine triphosphate, okay. There are 3 phosphate molecule that attach to the AD something, I forget, okay. There are 3 phosphate. Hello, don't move to the stream, real. Okay, do you bring your textbook today, or your textbook is in tong sampah? I hope my sound card will work well, okay. I charge it until green light, okay, not like Squid Game. Okay, if you notice the bottom left one, you will see some GIF, very funny GIF, okay. Esok sudah bagi cikgu, hai ya. No need, don't bagi cikgu, just buang in the tong sampah, okay. Screw textbook. Okay, so dark reaction. In dark reaction, or in another name is light independent reaction. Okay, again, light independent reaction, which this reaction does not need light, and this is the process where the food is done. Okay, where the plant make their food, okay. So at this part, carbon dioxide will be fixed, or we say in a very simple word, the carbon dioxide will undergo something and become food, okay. So this does not require any light. There is no light is required, okay. So the first one, light reaction, location is at thylakoid. So you ask me what is thylakoid, histamine, what is that, okay. In chloroplast, how to memorize them? You remember the casino chip, okay. Casino chip, stack, that is thylakoid, and light reaction happen here in casino chip, okay. So how the light works, there are called many, what you say, there are many details happening behind those thylakoid, which is super complex. And if I show here, my core current view will not be 0, and it become -5 or -50. Yeah, it's too complex. Anyway, light is involved during the process. Yes. How is it involved? They excite something in the thylakoid to make the thylakoid, something inside the thylakoid, to break down the water. Okay, to break down water. So lysis of water is important point here, so I cannot do anything. The lysis of water is like splitting the water. Okay, what does it mean? Water, as we know, is H2O. The photosystem, okay, photosystem in thylakoid will split, like you chop the water and give energy. Okay, you give energy through some process. Okay, they will chop the water and got energy. Impossible. They are going to do something process that too complex. Okay, so recap here. Light reaction is an energy generator for the photosynthesis process, which happen at thylakoid, and this process involve light. Okay, and what does it light do? It will split the water and generate ATP through the process. Okay, very short paragraph. The answer I gave maybe 7 out of 10 is correct. Okay, where is another 3 mark? The 3 mark will be the process. You need to explain. Paham, good. Okay, terima kasih. So yes, good. You learn something. Good. Okay, no quiz. Okay, in my lecture, no quiz. If you learn, you got. And then remember to revise back. If you don't revise, the knowledge will hilang right after I end stream. Okay? Tabun eh. Okay. So fun fact. Why most of the plant are green in color? Most, I say most, not the red in color or purple cabbage. I'm not saying that. I say the most of the plant you saw, the grass, the okra, the apple tree, the orange tree. Why is in green? Why they are green? Okay. So as we know, chlorophyll is a pigment. Okay, a pigment that eats different wavelength in light. Oh, wavelength. Okay, new words, wavelength in light. So let me read this. Okay, as a lecturer, we need to read slide and ask students to study themselves. For me, I don't ask my viewer to study themselves. Okay, but I still explain. Okay, so chlorophyll eats the different wavelength in light. Okay, so the chlorophyll are just a very general term for the pigment. Okay, for the pigment. Okay, just like human got Chinese, got Malay, got Indian. Okay, so that will be specified. Okay, so in specified form, you will have chlorophyll A, chlorophyll B, and carotene. These 3 are photosynthetic pigment in chloroplast or chlorophyll. Okay, blur juga. Tapi inside this have 3 main photosynthetic pigment, which their function is to absorb light. Okay, got it? If you got it, you say yes or something. Okay, because this part is like a key point. Okay, so what wavelength they eat? So we need to refer to this graph here. Okay, this graph. So what does this graph tell us? This is not stock value of any market. It just tell how much they eat at a certain wavelength. Okay, for example, if you see beta carotene, the wavelength is like this, up and a cat here and go down and habis here and does not go anywhere, which mean beta carotene, this pigment absorbs the wavelength from 400 to 530 something, which is, if you saw here, after my cursor go out, if you saw here, it absorbs purple, blue color and until a slightly of green color. Okay, what does it mean? Which mean this wavelength are gone, no more. Okay, beta carotene already eat. So what is left? Okay, so yellow, orange and red color is left. The beta carotene does not eat it. Okay, so what happened to the wavelength? The wavelength of the light, the color does not be eaten. So what is left is reflect 反射. It will reflect to our eyes and we see the color. So that's why beta carotene is orange or red color. Okay, this is how the lights work. Okay, whatever we see in this world is the reflection. Okay, reflection of lights. Very Okay, if you cannot understand again, let's try another one. Okay, we discuss chlorophyll A and chlorophyll B together. Okay, so what they eat? They also eat this one. Okay, they will eat the wavelength of purple, blue, and almost green, and then no more, but a slightly spike on this side. Okay, on the orange and red. So what is left is the middle part of the spectrum, the light spectrum. So what is left is reflected back to our eye. So comparing this 3-line graph here of this graph, the reflected back is green color until here. Okay, that's why the plants appear green. That means all things we see are fake. I won't give any answer on that. Okay, what is real is real. Okay, but fake, yes, could be fake because electron and electron never touch. They have a minor repulsion between electron and electron, so touch is never exist. We cannot touch anything. Just the feeling of repulsion of electron against each other. Hello Magic Cube, welcome to the stream. I hope you bring textbook and not poker card. How's you doing? How's you doing? How is everyone? Welcome to the stream. Welcome to the stream. What we see is like fake, right? Not fake. Very short lecture. I hope it's very short lecture. 30 minutes in, and we are halfway already. Yes. Live stream is just for me to like practice my presentation on this scientific topic, and some of you kacau me. I like it. I like my student kacau me. Okay, so for food assembly, or with another word, scientific term is dark reaction, or you want a technical term is like independent reaction. Going deeper, you'll be Kelvin cycle. Hate those friend who named Kelvin. Okay. No lah, just a joking. Kelvin cycle. Okay. If you learn science, you know lah. Okay. So where does it happen? This dark reaction. Location is a stroma. Okay. The spelling, if too long you teach the you teach the student science or something, if involve English, stroma and stomata. Stroma and stomata is different, totally different thing. Stroma with R is in the chloroplast, and stomata is located at the leaf where the air opening in and out, gases exchange happen there. Different thing, yeah. So don't get confused with the spelling. Stroma and stomata are different. Okay, back to here. Dark reaction, what do dark reaction do? So carbon fixation is occur here. So what does it mean? What is carbon fixation? So in a very short term is, or a very simple word is, chloroplasts use carbon dioxide and the energy from the light reaction. They fuse it together to become glucose. Okay. Or in a very simple word again, chloroplasts use energy and carbon dioxide. Pop, you become sugar. Our stomach is 肚子 different, totally different species. Good. Can think about 600. 600. I like this one. Okay, so in a simple, I don't go simple. Let's go a little bit technical. Carbon dioxide fixation or carbon fixation is almost different. Almost same lah. Okay, carbon dioxide also got C inside. It used energy molecule, which is adenosine triphosphate, to become glucose. Okay, so if you have some chemistry base, you will find out glucose is a 6 carbon compound, okay, inside the inside the formula of glucose, C6H12O6. This is the formula of glucose. Okay, so process does not involve light or photon. Okay, photon and proton lain is not the same. Photon is light, proton is the plus sign. Okay, so in this process, glucose is the main product and oxygen is byproduct. What does it mean? Okay, example, I'm chloroplast. I want to make food. I want to make food is the main point. When I'm making the food, something I made is glucose, yes. But the byproduct is like the sisa. Where like I accidentally create this one during when I doing this. Oh, very abstract. Okay, but anyway, you just remember glucose is main product and oxygen is byproduct. This is what that reaction do in photosynthesis. Very simple. Very simple. No light and you get food just like that. What is next? Let's see. Oh ho! You know what? 80% already done. So if I become a university lecturer, my lecture will be less than 1 hour. I don't know the topic. I don't want to choose big topic actually. Because if I choose big topic and then I stream for 2 hours, what I'm going to stream in my future? Experiment. Experiment I just like explain. Okay, what is this experiment? What is it for? If I want to do maybe in future lah, because this experiment involve alcohol. Yeah, involve alcohol. So how to get alcohol? The easiest way is the hand sanitizer. So I think what is the substitute? Okay, so no need PTSD, no PTSD. You know what my PTSD Every time I do experiment, my group, wherever group I go, I will be the last one go out from the lab. I don't know why. I always be the last. I don't know why. From secondary school, university also like that. Even from my first year until 3rd year, I always be the last one to get out from the lab. I don't know why. I already did it so fast. I did it correctly. Always be the last. I don't know lah. Okay, never mind. Okay, so in this photosynthesis, we have a mini experiment to study, not to study lah, is to determine what is the reason, what happened lah, what happened. So one of them are paper chromatography. Okay, chromatography is like separation of pigments using the paper method. So it is called paper chromatography. It have many different methods to do the chromatography. Okay, like the first point said, it is a method of separating plant pigment if used as plant. Chromatography are not only in photosynthesis but also in food, also in many other field of science. Okay, so the solvent used is a non-polar solvent. Okay, no plus minus. Okay, not water lah. Okay, what does it mean by non-polar solution or non-polar solvent? Okay, non-polar solvent is something does not ion. They don't have ion inside the solvent. Okay, if you don't know what is ion, the plus and minus, it will not form anything. Okay, so it will non-polar. Okay, although water, as we know, as we study in secondary school, water is universal solvent. Okay, universal. But it is not suitable to use in this experiment because it involves something like separating the pigment. Okay, so the solvent has to be organic solvent. Yeah, I remember like that lah, okay. But in here they use alcohol like acetone. Acetone alcohol lah. Acetone is alcohol. They use for the nail, what, 指甲油啊, for to remove that thing lah, 指甲油. Okay. So in this experiment is to determine the amount of pigment in plant. Okay. Paper chromatography is to determine the pigment, how much is the pigment inside the plant. Okay. Some have more keratin, some does not. Okay. Maybe because I do this experiment before in my STPM time, we use bayam hijau and bayam merah. Okay, so the pigment inside these two vegetables is not totally different. The pattern here is different. Okay, what the pattern you see here is 99% perfect. 99% perfect. And if you don't store it properly, the color of this paper chromatography will fade away. Okay, because it's organic and sensitive to light, it will oxidize and you get a very fade one, very dim. You cannot see the color already. Okay, so based on the result I search, okay, not me, this is not my handwriting. The pigment is here. Okay, you have keratin, you have xanthophyll, you have chlorophyll. Dark green is chlorophyll A. Okay, and then yellow green is chlorophyll B. Okay, so this experiment, what is the concept? If is you put the vegetable, you crunch, you blend it. I think it's blend it. You blend it until a very concentrated to break up all the cell wall using the acetone. Yeah, to dissolve it, not to dissolve. It's to break it down like very concentrated. Okay, and then put on one point here. After that, you dip this paper in the solvent until this level, like the blue dot here. Okay. After that, the solvent will go up by capillary action of the paper. It will go up suddenly. Yeah, you have to like hang it vertically, and the solvent will go up. As the solvent go up, it will bring the vegetable, like the condensed juice of vegetable of bayam. It will go up and separate the pigment. Okay. So the pigment with the small pigment, the small pigment with small molecular weight will go further. Those with bigger one, you move slow and then stain the paper here like this. So if the smaller will go further lah. Okay, so why we need to do this experiment? So based on my research, it is to study the photosynthesis efficiency of the plant. Okay, some plant have more chlorophyll A, some plant have more keratin, different. Okay, the second one is to study the stress response. And study me? Again, because when plant receives something like stimulus, it will respond through like making some chemical changes in the plant itself. Okay, so some of them may change. Okay, I need to produce more chlorophyll B. That one is another deeper level of study in botany. Okay, not agriculture. In agriculture, we need to know, okay, plant do photosynthesis and there are different type of plant pigment. Okay, the third one is to study the adaptation. So these two are related. Okay, they have stress and they need to adapt. How they adapt. The fourth one is biochemistry and or biotechnological application. So if the plant have more carotene, which means it has more orange or yellow color, it can use as food coloring, and some might extract as antioxidant. They undergo this study lah. Okay, a small experiment, but you can extract a much data or much information from the plant. Oh, I don't think I can. I never expect I can explain this much in this slide. Okay, this one even simple. Okay, a super simple experiment. Okay, it's called iodine test. What test? Test on starch. Okay, so like I said just now, this experiment is to study the presence of starch on leaf. Starch is 淀粉. Okay, why starch? Where is the glucose? Okay, so glucose is the basic one, or the simplest. Okay, simplest building block. Okay, so starch is a big chunk of molecule, you can say. Okay, polymer lah, we say. So hard to imagine. I don't know lah. The glucose you say is basic unit, and suddenly starch is a polymer. I don't know. Okay, imagine glucose is one piece of Lego, and you want to build a castle of using Lego. So what you build, you will stack those glucose together, and you stack, and become a big one, which is called the castle. You want to build a castle, right? So the castle is starch, which is the polymer, and yeah, that's the concept. You have monomer and polymer. Okay, so starch is the polymer. Starch is the polymer of glucose. So we can test the presence of this compound, starch, using iodine. How? You need to boil the leaf. Yeah, you need to boil the leaf to make the chloroplasts release all the content inside. Break down like your heart, like your ex break your heart. Okay, so break down, and then I forget the step actually, but the concept is there. You need to break down the chloroplast to force it to force it release the starch. So after that, you just use the dropper and drop some iodine on the leaf, and you become like this, dark blue. If the iodine change color from yellow, from this color to dark blue, it means starch is present on the leaf. Okay, so you might say, okay, yeah, I know, presence of starch, the leaf has starch. So what is so pro about this experiment? Actually, they do something like, okay, imagine here a leaf, right? On the top left of my slide, the leaf is here. If I cover a part of the leaf, and I put it somewhere. Okay, I put it somewhere because I cover already. So some of the leaf is exposed to light, and some of the leaf is not exposed to light. Okay, so they will study it lah. Okay, so the leaf will be boiled like usual. In this experiment, you boil like usual and then do iodine test. The result is the cover part will not change the iodine. It means when I drop it, it will remain the same color, the yellow color. But the part which is exposed to the light, it will become dark blue. So you will say that photosynthesis happen with the presence of light. So yeah, this one maybe I need to change for future reference. Okay. Oh, conclusion. Bangun from tidur. We need to end already. Wow, 1 hour. Ngam. Okay, so conclusion of this, more recap lah, not conclusion juga lah. Okay, plant make food using photosynthesis and light reaction generate energy, ATP. Dark reaction using energy to make glucose or further starch. Okay, so this reaction happen in chloroplasts. Light reaction and dark reaction happen in chloroplasts. Light reaction is at thylakoid, the carcinoid chip, and dark reaction is outside the carcinoid chip, which is the, I don't know. Yeah, dark reaction is outside the thylakoid. Okay, inside chlorophyll. Inside chloroplast. Sorry, inside chloroplast. Okay, so by doing this, okay, light reaction, dark reaction, what's the point? So it is like it help us to understand plant grow, farming and gardening. So it is important lah. Okay, light reaction. Where do you need to put the plant? Also, question. Okay, I shall put it under open field, or do I need to give any shade to the plant? Yeah, it's a question to consider. So it brings us to the question below. If lights help plant to grow, what if there is too much light or too little light? So for some plant, some plant like open field because they receive much light. Some plant are introvert, they don't like to get exposed to the light. They are more to shade. They go shade. Okay, you need to give the plant a little light. Put it under a roof or inside your roof. Okay, some plant like this. Okay, what happen if you put the shade level? Okay, we say shade level to open field. Definitely you're like, oh, cannot, I get burn. So you get sunburn, or you get burn mark on the leaf. Okay, so it's just like you pressing your plant too hard, and then you become like, oh, I don't know, I gave up living. So the plant will get stressed if it's supposed to be in shade, but you put it under a big hot sun, like that. So the balik, if you put those plant would like sunlight and you put it indoor, if you're stressed also, because you are how to say, just like you don't give the children go outside. Oh no, teacher, I missed the class again. Never mind. You can watch the VOD again. No. I know you are at somewhere else. But I don't blame you. Okay. Okay, so the question is like, can hit to the next live stream actually, because some gardening tips also involve photosynthesis. Like, okay, this plant need more light. Okay, where should I put? This plant need less light. Where I don't put? This is another topic to discuss in gardening tips. So for now, maybe more on plant science first before we go to practical. University syllabus. Let's test this. I hope it's not too loud. Okay, so yeah, this is end already. So yeah, the best slide you can see in university lecture is these two words: thank you, or you want terima kasih. Yay. Yes. Oh, end already. Yes, histamine stream is always short and short and simple. But if you saw the content, it's very hard. STBM. It's very easy only lah. Sumpah easy. Because STPM already tell you sijil terlalu paling mudah, and SPM is sijil paling mudah. Sijil paling mudah. SPM sip suay lah. I can pass. SPM I can pass easily with 2A. 2A only, I tell you. Mathematics and chemistry. Okay. No need to like, okay, you go straight A, you must straight A. I pass my BM, I pass my sejarah, and English can use lah. Okay, English can use now like this broken. You say broken English, not broken juga. Okay, we Roger English lah. Okay, Roger English you can use in Malaysia. My BM okay lah. Okay, but the problem is luckily my MS is E lah. E, elephant the E. Okay, if F already, say bye bye to STPM no need to go lah. And then my STPM also easy. Okay, STPM science stream I got only one A minus that is smaller, which means you are a educated person with big moral. Big moral. I don't think my moral get A, or my moral get B. So this proof I'm not an Asian. So Tulang, where is your result? You become a teacher, I need to know the result. Comment down your result, or you get 600 instantly again. Your mathematics G? Your mathematics G OG. Oh sorry, okay. No, don't what we call, don't 言上me. Forget Sudan, maybe I want A for math. This bro is confirmed 100% Chinese because good at math. 1 plus 1 is 2 minus 2 plus 2 is 4 minus 1, that's 3 quick max. MS is not a subject for human, that's a subject for aliens. We know, at least we know y equals mx plus c, and 1 plus 1 is 3. Good. Now what is done. Okay, we chill with our job now. We got food to eat. Okay, it's good lah. So Tulang 老师, can you share my stream to your dear students? So they got extra class, so no need to go extra class. I'm not joking. No need to share. So when will 1 plus 2 not equal to 3? You put, I don't know lah. In your math is like, screenshot. Let me screenshot. What the hell? What the hell? When will 1 plus 2 not equal to 3? Aduh. 1 plus 2 not equal to 0, none. I don't know, man. I am not Chinese. My Chinese is not Chinese thing. I cannot do mathematics. Can we do mathematics? Can. Agriculture got mathematics. What is the mathematics in agriculture? Agricultural economic. I got B on that subject, and I don't know what I've studied. Agriculture economic. Let me tell you lah what I learned. Let me show my Instagram. Hey, I think we can do some zatan. Okay, good. It's my agricultural meme. I already doing the content for tomorrow's stream right now. tomorrow's stream is about stereotype of agriculture.刻板印象 at 2 p.m. Okay, so we do some jatan. Okay, since you two are still here, helping my watch hour. Thank you so much. What to do? With the window capture, we do some window capture. If you notice down here, here got the gift. 猴子在这个世界上没有谁能够审判我。Jatan first. Okay. Maybe until 10:30 or 10:30 we off. 10:30 we off. Okay. 1 hour class. Okay. We still got 5 minutes to do something stupid. Instagram one more. Yes. Yang Hydrate tuition. Do you know what is the best part of in the class when the teacher teach some subject is boring? The teacher when the class how to say? The class will be more interesting if the teachers share something unrelated to the subject. Unrelated. Okay. Do you know? I divorced with my wife. Okay, everyone gets attention. Okay, so we're going to do something different. Okay, so this one. When you are dead and you are realize you don't have time because you have to study genetic and animal breed. Not relatable. But my teacher don't want to do that. Oh, bad teacher. I'm a good teacher. Comment, subscribe for more teacher content with not related to academic. The best tuition teacher ever. At least we learn something. Yes. Not learn something bad. Good. You are not learning how to make drugs. Okay. How to make what? How to make weed. How to plant weed. Okay. 没有教你种大麻. Oh. This one not relatable. What is this? Weed. Another weed. This one I don't know lah. This one the what we call this one is for the livestock production. About livestock. Yeah. So the only warmest play on farm is this inside the butthole. Some almost wake up at 3 a.m. to make sure others have milk at 9 a.m. Unrelatable. Yes. They do this. They do this in farm to check. I think it's to check. I don't know lah. Because I'm not into the livestock stuff. Either they check the cattle inside 小牛 inside the mother cow there, or they're playing with the gastric. No, the digestive system, I think. Yeah, something related to their healthy. Yeah, because as we know, not as we know, as I know, cow have 4 stomach and different functions, so they need to feast the cow to check something lah. Okay. Imagine when you okay, I'll stop. Never mind. If you want to continue, you continue. I don't force you. I won't force you. As always, I start the struggle of study animal science. Actually, I not study animal science. Me when I found out there's soil science teacher and absent. I don't know why they're so scared of soil science. Back when I am first year, I studied soil science. The lecturer was so chill. And it does not the the content is not that hard at the first part because it's just introduction, and then soil science, the second part, which is super terrifying because it involves so much of chemistry that I never see before. Yeah, soil science is a very big field to study. Okay. This one is a variety of pig, variety of babi. We got large wild this. What the fuck? Muller food or kind. I never see this before. Okay. So much animal. I think people study animal science is more stress than plant science. No sound already. Ay. Got. Hush. Oh. Sound blinking, which means I need to off already. Oh, okay. I know the half-life. No job. Oh. Okay lah. Tomorrow stream will be something like this lah. Okay. I made a form to ask feedback like, okay, what is your stereotype about agriculture? Zero response. So okay, I need to switch strategy. I need to review this beam here, everything here for half an hour or 1 hour. So so what? Terpaksa lah using another mic. Luckily I got mic don't have far. Okay. So we need to end stream now because my mic already dead. So I think that's all lah for today. Okay, we already do something like about the photosynthesis and some beam review on this Instagram page. Okay, so let's end stream. So bye bye everyone, bye bye Tulang, bye bye Kawi, thanks for coming, and bye bye Magic Kim, happy Didi. And that's all for tonight. Bye bye. Time to go our there. Okay, go our there. Bye bye. Arigato.
Close to the sun.